I'm talking about small cell lung cancer, some new targets, some new strategies, and some updates that came out of our most recent big oncology meeting. So what do we want in small cell lung cancer therapy? Well, of course, we want it to be effective. We want it to shrink the cancer and help people. We want it to be specific to the cancer cells. We don't want the therapy to affect the normal cells in our body, because that's where the side effects come in. We want it to be really toxic to the cancer, but we don't want it to be toxic to the person with cancer. And we want it to work for a long time. So small cell lung cancer arises in the lung. So on the left-hand side here, we have a cartoon of the lungs. So kind of the bluish thing coming down is the trachea. It's the windpipe, and then it divides and goes into the lungs. It's kind of like an upside down tree. And in those airways that are within the lungs, there are cells that line the lungs that we call epithelial cells or lining cells. Some of them are neuroendocrine cells. So these are the cells that line the lungs. And they're the cells that tend to get exposed the most to carcinogens that we inhale. And in the small cell lung cancer, that usually is exposure to tobacco smoke. And that makes up about 90 to 95% of what we'd call small cell lung cancer. There's a small percentage of small cell lung cancer nowadays that comes from transformed EGFR mutated lung cancer, which is kind of a special case. And the fact that we talk about smoking history is just that's the biology of it. In all of lung cancer research and advocacy, we have to get rid of smoking as a way to criticize or stigmatize people. You know, both my parents were smokers. They were good people. My dad served in the Navy and in Korea. My mom worked at home, raised 4 kids. I'll let you decide who did the most combat time. So anyway, the cells that line the lungs get exposed to carcinogens, and they can transform into different types of cancer. When cells in the lung transform into cancer, about 15% of the time now, it's into small cell lung cancer versus the other varieties. There are a number of different molecular changes that are very specific to small cell lung cancer. Up at the top, you see RB1 and TP53. These are genes that are almost always mutated, or function is lost as cells go from normal cells into small cell lung cancer cells. And these molecular changes are kind of very specific and change a lot of different production of proteins in the cells. And these are really what is going to lead us into the future to be able to target the small cell lung cancer. The fact that these cells transform, they change, and they start to produce some proteins on their surface that are not produced very frequently in normal cells. So what about traditional small cell lung cancer therapy? So, chemotherapy has been the mainstay of small cell lung cancer treatment for many decades, and it remains so. So the most common chemotherapy combination in the United States would be either carboplatin and etoposide, or cisplatin and etoposide for upfront treatment. And chemotherapy is used in basically all stages of small cell lung cancer initial therapy. Radiation is really important. And that is really important for limited stage small cell lung cancer when patients present with cancer that's limited to the lungs, and we give the radiation with chemotherapy to try to cure the cancer. And we've had that approach for about 30 years. We can also use radiation therapy to treat cancer when it goes to the bones and is causing pain, or when it goes to the brain and we're trying to shrink the cancer spots specifically in the brain. Over the last 10 years, we've incorporated immune therapy into the treatment of small cell lung cancer. And what's really exciting about that is that adds to the long-term control of cancer. Because when we get the immune system going against the cancer, there's the potential for it to control the cancer for years. So that's really exciting. So how does the immune system work? Mostly when we're talking about immune therapy in lung cancer, we're talking about T cells. So the T cells are immune cells that have the potential to recognize proteins that are not made under normal conditions in normal cells in the body. And I have, if you heard me talk at the meeting that we had in May, then you may have heard me to refer to them as kind of like the Pac-Man of the body, where those little mouths that kind of go across the computer screen trying to gobble things up. The T cells are kind of like the Pac-Man of the body, and they roam around looking for something that doesn't belong to attack it and essentially eat it. But there are a lot of reasons that those T cells can be turned off or fail to activate. One of them is when the tumor cell produces a protein called PD-L1. And basically, it can flip the switch off on a T cell. And on the left-hand side of this panel, that shows where the T cell is being turned off, because the PD-L1 interacts with the negative checkpoint PD-1, and that short circuits the T cell and turns it off. On the right-hand side, if you look at the little yellow shape, that shows an antibody, and an antibody like that would be like Keytruda or Opdivo or Libtayo that binds to the PD-L1 or the atezolizumab or durvalumab, which is in Finzi and Tecentriq. So when we give somebody an antibody, one of these immune therapies intravenously, that protein floats around in the body and then binds to one of these receptors, prevents the T cell from being turned off, and then the T cell can attack the tumor cell. Okay, I think there was a raised hand. Does someone have a question? Is Wendy?
Thank you. I did want to know, we always hear about these T cells, but what does the T stand for, and where is that located?
I think that it means thymic.
Okay.
Like derived from the thymus.
Okay.
When T cells form before we're born, in an organ called the thymus that is up around where your Adam's apple is.
Okay.
Okay. And kind of a miraculous things happens in the thymus where these early T cells generate billions of different T cell receptor variants for some miraculous process that is way beyond my ability to understand. The body takes any T cell that reacts against ourselves and it destroys 99% of those. So if the T cell is going to attack us to cause rheumatoid arthritis or lupus or colitis, psoriasis, the body can eliminate them. If it's a lousy T cell, if its T cell receptor just doesn't recognize anything, it's just kind of blah, those get eliminated. And that leaves us with billions of different T cells that have the potential to recognize things that don't belong, like a virus, a bacteria, a parasite, a virus infected cell, and in some cases, cancer cells. Most of the time in treating cancers, the cancers that are likely or more likely to be susceptible to immune therapy, it's when the cells are very different than normal cells. So cells that have undergone a lot of mutations in their DNA, exposed to a lot of carcinogens, like with tobacco smoke, exposed to a lot of sun exposure. So some of the skin cancers like melanoma, Merkel cell, and cutaneous squamous cell cancer are really susceptible to immune therapies. And we think it's due to the fact that they have like a gazillion mutations in them that produce a lot of different abnormal proteins that you have a good chance of having T cells that will recognize an attack. Okay?
Thank you.
Yep. So we talked about there are these PD antibodies that are made. Clever scientists can get cells basically in a test tube to make antibodies against the off switch on the T cells. So we can identify, we can cause these antibodies to be generated that block the off, that glom on to the off switch. And the way I explain it to people that I see in the clinic is I say, imagine if you could just take a big piece of duct tape and put over a light switch. Now you can't turn the light off. Okay. Same thing with these antibodies. We put them on the off switch of the T cells, and now the cancer cell can't turn them off. So that's kind of the basic mechanism. And it takes the brakes off the T cells so that they can be active and go after the cancer. Small cell lung cancer often has these protein targets. Small cells often have a lot of mutations in them and generate a lot of abnormal proteins. But for reasons we don't fully understand, the small cell lung cancers often have a microenvironment. In other words, in the tumor, it's not a good environment for the T cells to work, which is why we don't see as many immune responses as we might like or might expect, given that small cell lung cancer arises from tobacco exposure, is a rapidly growing cancer, and has a lot of these mutations. But it does work, and it's been a big breakthrough. But there are still mysteries that remain around small cell lung cancer, like why don't we do better with immune therapy in small cell lung cancer? So I'm just going to show you one slide each for some of the established therapies. So for patients who present with metastatic small cell lung cancer that have had no prior treatment, based on a couple of big trials, we now treat with standard chemotherapy plus immune therapy. So this is the Empower 133 study that showed that carboplatin, etoposide, and the immune therapy atezolizumab gives better overall survival compared to chemotherapy alone. So that helped that get FDA approved and become one of the standards of care. This is the Caspian study. And it showed that chemotherapy plus durvalumab gave a better survival than chemotherapy alone in extensive stage small cell lung cancer. So that's why chemotherapy plus durvalumab is one of the other standards of care. So there are two good standards of care for small cell lung cancer, and a physician could recommend either one, and they're both good treatments. At ASCO 2024, there was a new therapy identified in early stage small cell lung cancer. So patients that we have traditionally treated with chemotherapy and radiation to try to cure the cancer. And this showed that if we gave chemotherapy plus radiation, and then afterward gave the immune therapy durvalumab that we just saw earlier in the Caspian trial, you can see they had this theme where they named all their trials after oceans or seas. And in this trial, if people got durvalumab after chemotherapy and radiation, there was a substantial improvement in both progression-free survival and overall survival. So a very exciting new treatment to offer where we anticipate that we'll be curing more people with limited stage small cell lung cancer. So what about new therapy in small cell lung cancer? Well, we do have new chemotherapy that works through slightly different mechanisms and now has found a new place as part of first-line treatment. We have antibody therapy alone, where we make monoclonal antibodies that target specific small cell lung cancer molecules. We have antibody drug conjugates, where you make an antibody against a protein on the small cell lung cancer cell, and you glom a really toxic chemotherapy onto that antibody, so So the antibody delivers that nasty chemotherapy directly to the cancer cell, sort of targeted chemotherapy. We have bispecific antibodies now, where the antibody gloms onto a protein on the T cell and onto the small cell lung cancer, pulls them together to make an immune attack by the T cell more likely. And then CAR T cells is where they take a T cell, kind of strip it down, take off all the brakes, put on some more gas pedals, and it just goes. And we'll talk about that also. So again, we kind of go back to small cell development and just emphasizing that we get all these molecular changes that lead to the production of new proteins that are not present on normal cells that show up on the small cell lung cancer surface. So this is a cartoon of a cancer cell, in this case, a small cell lung cancer cell. In the middle of the cell, you have the nucleus. That has the DNA. So that's the genetic material. That genetic material codes to another nuclear type protein, RNA. And then the cell reads the RNA, puts amino acids together, and that makes a protein. And often the protein will get stuck on the surface of cells. When there are mutations in the DNA, that can cause abnormal proteins, but it can also cause proteins to be produced and expressed that aren't produced on normal cells. They're only produced on the cancer cells. So here are 5 important ones. DLL3 is already a target for approved treatment for a bispecific antibody called tarlatamab that I'll talk about in a few minutes. B7H3 is another target that is already under advanced clinical trials for an antibody drug conjugate against that. TROP2 is present on small cell lung cancer often and a lot of other cancer cells. So TROP2 antibody drug conjugates were approved in some other cancers like breast cancer and maybe soon in non-small cell lung cancer, and has shown promise in small cell lung cancer. SEZ6 and fucosyl GM1, where there's an antibody, just a bare antibody against that protein that has shown promise. And there are antibody drug conjugates coming for that also. So what does an antibody look like? So antibodies are proteins made by immune cells called plasma cells. And these immune cells called plasma cells are very similar to T cells. But what they do is they have a receptor that identifies, that is coded to identify abnormal proteins. And when they see them, they start to make the antibody that binds to that protein, and they spew it out into the blood. So you can get these really high levels of these immune proteins in the blood. And when they glom onto a cancer cell, it targets that cancer cell for killing by T cells and macrophages and natural killer cells and other parts of the immune system. So the antibodies produced by the plasma cells are really important for fighting infections. And in this case, they can be really important for fighting cancer. And now that we have really advanced science, really clever scientists are able to do designer antibodies where they can actually look at the proteins on a cancer cell, and using advanced computational modeling, they can predict what the antibody needs to be to bind to it. And they're able to cut and paste antibodies together to make antibodies to go after the targets they want, and to make antibodies that hit more than one target, where we're getting these now bispecific and trispecific therapies. So some of the ways that we can use antibodies And similar approaches are with antibody monotherapy. In this case, a drug called atototog that I'm going to talk about. Antibody drug conjugates, where we have antibody with chemotherapy. Bispecific antibodies, where the antibody gloms onto a T cell and also gloms onto the cancer cell and pulls them together. And trispecific antibodies, where that antibody gloms onto the T cell, but then basically can grab on to the cancer cell with two arms and really get a good hold on it, okay? And then CAR T cells. So here's a picture of an antibody, and there's an antibody that's made against anti-fucosyl GM1, and this is a protein that is expressed on the surface of neuroendocrine cells. In other words, cancer cells that have neuroendocrine characteristics, which small cell lung cancer is sort of the prime example of that. So small cell lung cancer, 70 to 80% of the time has the fucosyl GM1 molecule on its surface. And an antibody developed in the laboratories of Bristol Myers Squibb has been studied along with the Immunotherapy Opdivo, a PD-1 checkpoint antibody, and combining the antibody with the Opdivo showed promising results in previously treated small cell lung cancer. A randomized phase 2 trial looked at chemotherapy and the Opdivo with or without the atototog, and the overall survival was better with the addition of this new type antibody. And now there's a large international phase 3 trial being done throughout the world comparing chemoimmune therapy with or without this antibody, this atototog. These are some of the antibody drug conjugates. So we talked about the DLL3, the TROP2, the B7H3, the SCC6, the fucosyl GM1. And what these are is, if you kind of look up at the top where it says general structure of ADC, it shows an antibody that has the potential to latch on to a cancer cell. And those little yellow six-pointed stars represent really toxic chemotherapy molecules. You give this intravenously, it goes through the body, it finds the cancer cell, and then down on the bottom, it shows those molecules going onto the surface of a cancer cell. It gets sucked into the cancer cell, the chemotherapy gets released, and it poisons the cancer cell like a poison pill, okay? So there are about 4 or 5 or more antibody drug conjugates in advanced clinical trials in small cell lung cancer. Some of them have looked quite promising. So this is a line of treatment that is coming along. And then we have bispecific antibodies. One of the bispecific antibodies, tarlatamab, is already being used in the clinic based on the results of phase 2 trials. So tarlatamab is an antibody that can recognize 2 different things. It can recognize the CD3 protein that is always present on T cells, so it can latch on to the T cell, but it can also latch on to DLL3. That's a protein that is expressed on about 70 to 80% of small cell lung cancer cells. And what it does is it just pulls the T cell and the cancer cell right next to each other. Like they're only like 1 molecule apart, just nanometers apart. So they're cheek and jowl, they're right next to each other, okay? And that way the T cell is kind of roped right next to the cancer cell. And if it has the ability to recognize and attack that cancer cell, it greatly increases the chance for that. And then that T cell attack can potentially kill off the cancer cell. This shows one of these fancy CAR T cells. So if you look up in the middle there, that blue T cell, it has had the brakes taken off. So like the PD-1, which could be inhibited, that's been removed. The T cell receptor has been revised so that it doesn't need other activation ways. So for these T cells, we don't want them to attack when they shouldn't. We want them to be neutral. Under normal conditions, we want them to just sit there and mind their own business and do nothing. So there are about 5 or 6 different breaks, and there are also about 5 or 6 different activation levers or switches on them. And all of those things can inhibit the T cell from attacking a cancer cell. So when you're designing a cell to be able to just attack the cancer cell with reckless abandon, you take off the brakes and you combine all the activation switches together and bake them right into the T cell. So when you infuse those T cells into the body, they just go to the tumor cell. There's nothing to stop it, nothing to put the brakes on it, and it attacks. Now, the problem is that can sometimes lead to the T cells running amok and attacking other parts of the body. So there are lots of really clever ways to get around that. One is that sometimes the T cells will have a limited life expectancy. So they have sort of a kill switch in them that after a few days, they just all die. So they come in, they attack a bunch of stuff, and then they go away so that you don't get a chronic treatment-related immune toxicity. So, you get the impression that this is very complicated. It is. And there are a lot of challenges to making it actually work. There are, but there are also many possible advantages, and it's very exciting. And there are already clinical trials in process looking at CAR T cells against DLL3. So, it's not ready for prime time. You're not going to be offered it as standard therapy in your doctor's office in the next year, but it is already in the clinic. So it's not just some dream. It's being done. And these CAR T cells could come into treatment for different lung cancers in the not too distant future. CAR T cells are already part of standard of care in some of the blood cancers. So it is already FDA approved in certain lung cancers, in certain blood cancers, but in some ways some of the blood cancers are simpler because some of them have proteins that are only made on those blood cells. They're not made on anything else. So it makes it a simpler sort of targeting problem. So what about ASCO 2025, our big meeting first weekend in June every year in Chicago, because it's one of the only cities in the country that's big enough to host the 40 or 50,000 in hotel rooms. So there were 2 trials that are practice changing, and then one that's just sort of exciting and new. First, I'm going to talk about the IMforte trial. So lurbinectedin is a new type of chemotherapy in small cell lung cancer. It has been preliminarily FDA approved for the treatment of second and third line small cell lung cancer, so after initial treatment. But the folks that were developing lurbinectedin said, well, why don't we try it in first line therapy as maintenance? So after people have received 4 cycles of standard chemotherapy with carboplatin and etoposide, in this case with atezolizumab, patients who were doing well after the chemo were randomized to either continue the atezolizumab, which is the standard of care, that's what you would get off of a clinical trial, or to get the atezolizumab with the lurbinectedin chemotherapy that works through a different mechanism of attacking the cancer cells than the first types of chemotherapy, the carboetoposide. So it's either patients got standard treatment and then were randomized to either get standard immune therapy or the same standard immune therapy with a new type of chemo. I like to show the toxicity first because it's really important for us to realize that toxicity is important. Side effects are important. Quality of life is important, of course. In this trial, the addition of the chemo did add some increased toxicity, and it's the toxicity of chemotherapy with some nausea, some low blood counts, some fatigue, but it did not add to the atezolizumab toxicity in a dramatic or unexpected way. So yes, more side effects, but not dramatically so. And this is what we call a tornado plot. So on the left-hand side, the blue bars are for the experimental therapy with the lurbinectin and the atezo. And on the right-hand side, kind of the red and the pink, that shows for the atezolizumab immune therapy alone. It did increase toxicity, the chemo toxicities. But it did improve overall survival. So this shows a survival curve with about a 3-month increase in overall survival with the addition of lurbinectin. So I think most of us felt that this was not only a statistical improvement, but a clinical improvement. As a chemotherapy, we generally expect that this will not markedly increase long-term survival at 3 and 4 and 5 years, but definitely a step forward and a way to improve treatment in the here and the now. So we think that this was clearly a positive trial and will become an important option for patients and their physicians. And this is just the conclusions that I just kind of went over, showing a significant improvement in both progression-free survival, in other words, the likelihood of the cancer coming back. And probably most importantly, there was a improvement in overall survival. I'm going to talk about another trial called the Delphi-4 trial, and that was with tarlatamab. That's the bispecific antibody I discussed a little bit earlier. And this is in second-line treatment, where patients had standard treatment with chemotherapy and immune therapy, and then the cancer unfortunately came back. In this trial, patients enrolled and then they were randomized to either get the standard of chemotherapy that they would have gotten anyway outside of the clinical trial, or the tarlatamab bispecific antibody, the antibody that gloms onto the T cell and to the small cell lung cancer by latching onto the DLL3 protein. The side effects of the tarlatamab are to cause an intense immune reaction, and this is what we call cytokine release syndrome, and that can be with fevers and chills and wheezing and shortness of breath and low blood pressure and other side effects. So significant side effects and ones that have to be considered and particularly considered based on a person's specific other medical conditions. Tarlatamab versus the standard chemotherapy showed a significant improvement in overall survival of about 5 months. So a meaningful improvement in survival. Because this is an immune therapy, we're relatively confident or optimistic that this is going to lead to improved overall survival long term. So improving survival at 2 and 3 and 4 and 5 years. So certainly an important trial. This is a tarlatamab trial that was done a few years ago and presented. And I just show this. This is called a swimmer's plot, where each of these blue lines is a different patient. And down below where it says 10, 20, 30, 40, 50, those are weeks on therapy. And if you look out at 50, which is about a year, many patients are still alive with a response and with getting ongoing treatment. So we really think that the tarlatamab is going to produce prolonged improved outcomes. So the tarlatamab in the second-line setting produced better overall survival. Side effects are significantly, but usually manageable. And this really provides potential for prolonged cancer control. This is sort of exciting new technology with a trispecific antibody. So this is an antibody that grabs onto the CD3 and then grabs onto the DLL3 in 2 different ways. So this is a molecule that can grab onto the T cell and then two different places on the cancer cell, really grabbing onto that cancer cell with two hands. And this is a cartoon showing that. Green is the T cell, the immune cell. Pink is the cancer cell. And in between, it shows that trispecific antibody latching on to both the T cell in one spot and the cancer cell in two spots. This shows some of the side effects with significant cytokine release syndrome. And this shows some of the responses. So about 60% of patients who had had previous immune therapy had more than 50% tumor shrinkage here. You can see that in the green table on the left-hand side, where PR means partial response. That means at least 50% response shrinkage of the tumor. And then over on the right-hand side, that's what we call a waterfall plot. I apologize, those numbers are a little bit low, but that line in the middle with the zero, let's see, there we go, I have an arrow, good. If you look at my arrow there, that's going along the zero line. What that means is the size of the cancer when treatment was started. Okay? Each bar is the size of the cancer for an individual person on the trial. If the bar goes down minus 100, that means it's 100% shrunk, a complete response. Okay? If it goes up 100, that means the tumor has doubled. It's 100% bigger. Okay? If it's zero, it means it stayed the same. Here you can see that almost all the bars are below the line, many of them way below the line, showing that with this particular treatment on this particular trial, the vast majority of the tumors shrank. So this is a promising preliminary result. And this therapy is ongoing in different clinical trials at this point. So the conclusions are that this is an exciting new technology giving good preliminary results, and this is going to lead to additional clinical trials to see where this may or may not find a place. So conclusions for 2025 in small cell lung cancer right now. They're exciting results, but we're not where we need to be for patients and their families. Until we're controlling the cancer for a long period of time for almost everyone, we can't stop and rest and congratulate ourselves too much. But now there's hope. Particularly with the addition of immune therapy, there's the potential for just about everybody that they may live a long time and do well for years and years. So for somebody that's was doing lung cancer research before I could say that's exciting to me, that really we can offer everybody hope. All of our current treatments can produce prolonged survival. So everybody has hope for living a long time and doing well. The new therapies, particularly the ones that are targeting the immune system, are improving outcomes. Targeting these specific molecules in the small cell lung cancer space, the ones that we talked about, DLL3, SEZ6, the fucosyl GM1, all of those molecules really are leading to breakthroughs. And these multiple new therapy approaches, both as first-line treatment, second-line treatment, and in the treatment of early-stage disease, I am confident are going to move things forward in a real way in the near future. So thank you all for your attention, and I appreciate the chance to talk to you guys.